PCR in molecular paleontology. Paleontology, it mainly involved the study of some ancient living things, and molecular paleontology, it mainly refers to the recovery of the biomolecules, like the recovery of the DNA or protein or other materials like lipids, carbohydrates from the fossils, or some human skeleton that are very ancient, or some other buried materials of animals or even plant fossils. So it is called as the molecular paleontology, and the DNA or protein sequence that is recovered, it can be used to find the similarities of the ancient living organism with the organism now inhabiting our earth. जो फर्स्ट इंफॉर्मेशन थी जो यूज़ की गई थी, या जो मॉलिक्यूल जो कि एक्सट्रैक्ट किया गया था फॉसिल से या किसी और सैंपल से विच इज़ वेरी एंशियंट or that has been recovered from some archaeological site like human old skeletons or some other animals. जो फर्स्ट एविडेंस थी किसी लिविंग ऑर्गेनिज्म से, that was around in 1950s when some amino acids they were recovered from the fossils or some ancient samples, and then the amino acid sequence it was used to elucidate the relationships among living organisms. इसी तरह से with the passage of time, DNA it was also extracted from some ancient samples or fossilized samples. Now the problem is that when we are going to recover the biomolecules from the fossils or some ancient materials, the problem is that the genomic DNA or nuclear DNA, it can be degraded very rapidly as soon as the death of the organism is there. When death is there, the nuclear DNA is degraded very rapidly. And then people focused on the mitochondrial DNA or the DNA from the chloroplast, which is relatively stable. So, but still problem is there that during the extraction process, the quality of the DNA, it is lower as compared to the nuclear DNA. But still, people are able to extract the information from the ancient sources, and sometimes it is also called as the ancient DNA. So this technique, that is PCR, it can also be used to get information from some fossil record. So far we have discussed that what are PCR, what is the basic reaction, what are the basic variations of the PCR starting from multiplex PCR, RT-PCR, qPCR, or competitor PCR, inverse PCR. So we have discussed a variety of type of PCRs, and today we also focused on some applications of the PCR.